podcast from BioWintron, where we bring you the latest antibody industry trends. This month in June, we'll highlight the era of AI-driven antibody design. Artificial intelligence can now help researchers analyze antibody sequences, predict structures, prioritize mutations, optimize candidate properties, and most excitingly, generate entirely new antibody sequences. These capabilities are changing what can be proposed computationally. But every proposed sequence raises a practical question. Will the antibody actually work? It must first be produced, it must fold correctly, it must bind the intended target. It may also need to demonstrate suitable specificity, purity, stability, and other properties relevant to its intended application. That transition from computational design to experimental evidence is the focus of BioWintron's new Rush Data service. Rush Data is designed to support AI and machine learning-driven workflows by rapidly producing and testing large panels of antibody candidates, then returning actionable experimental data. First, what exactly do we mean by AI-driven antibody design? An AI model might identify patterns within antibody sequence datasets. It might predict the three-dimensional structure of an antibody or model its interaction with an antigen. It might rank mutations intended to improve affinity, suggest changes associated with humanization or developability, or generate new candidate sequences based on a defined target or design objective. AI-assisted antibody discovery may begin with experimentally identified antibodies and use computational methods to prioritize the most promising candidates. AI-guided optimization usually starts with an existing antibody and proposes sequence changes. De novo design is more ambitious. It attempts to generate a new antibody or binding molecule for a specified target. One example is Chai Discovery, a company worth $1.3 billion, with which both Pfizer and Eli Lilly have executed major licensing deals. Their system, Chai-2, achieved an overall hit rate of about 15.5% for de novo antibody designs. Almost half of the tested targets produced at least one experimentally validated binder, generally after testing fewer than 20 designs per target. Their next model, Chai-3, shows step-change improvements in AI-driven antibody design, doubling the success rate of Chai-2 and producing antibodies that meet required therapeutic standards. The company described this as more than a 100-fold improvement over the hit rates reported for earlier computational approaches, suggesting that de novo design may be moving from large, low-yield computational libraries towards experimentally manageable candidate panels. Machine learning systems are influenced by the datasets used to train and evaluate them. In antibody research, those datasets can be incomplete or uneven. Large sequence databases may not contain reliable heavy and light chain pairing. They may lack quantitative affinity measurements, epitope information, expression data, negative results, or consistent assay conditions. Data gathered from different laboratories may also be difficult to compare. Differences in expression systems, antigen preparations, assay formats, concentrations, controls, and data processing methods can introduce variation that has little to do with the antibody sequence itself. For an AI workflow, a smaller set of carefully standardized measurements can help. As computational systems become able to propose more candidates, producing and comparing hundreds or thousands of physical antibodies remains a challenge. The scale of industry interest is reflected in current market projections. According to Research and Markets, the AI in antibody discovery market is estimated at approximately $529.9 million in 2025 and projects it could reach $4.84 billion by 2035, representing a compound annual growth rate of 24.76%. These figures indicate strong expectations that deep learning, generative AI, multi-omics integration, and computational design will become more deeply embedded in antibody research. The broader outsourcing environment is also expanding. The global pharmaceutical CRO and CDMO market has been estimated at $254.65 billion in 2025, with projections suggesting growth to approximately $594.07 billion by 2035. Taken together, these projections point towards increasing collaboration among computational biotechnology companies, pharmaceutical developers, and specialist and experimental service providers. Another example of an AI discovery platform is Nabla Bio's Joint Atomic Modeling or JAM platform. Nabla used JAM to design antibodies against multipass membrane proteins, including Claudin-4 and CXCR7. These targets are difficult for conventional antibody discovery because only limited regions are exposed outside the cell. Their extracellular loops can be flexible, and closely related membrane proteins may present substantial specificity challenges. The company reported antibodies with affinity, epitope specificity, functional activity, and developability-related properties without a conventional optimization campaign. It also described a form of test time scaling in which allocating more computational effort during design improved the probability of obtaining successful candidates. Similarly, the company Absci has built computational design and experimental screening into a closed-loop system. Its integrated drug creation platform uses GenAI to propose antibodies at selected epitopes and then feeds experimental results back into subsequent modeling. The investment also shows how seriously the industry is taking this model. Zyra Therapeutics launched in April 2024 with more than $1 billion in committed capital. The company combines machine learning, biological data generation, and therapeutic development, drawing on protein design technologies associated with methods such as RF diffusion and RF antibody. So are we now in the era of AI-designed antibodies? Well, the more accurate description may be that we are entering the era of experimentally integrated AI antibody design. The competitive advantage will come from learning more efficiently from each sequence that is built and tested. That requires computational models, but also scalable antibody production, well-designed assays, standardized data, and rapid feedback. And that brings us back to how we can help AI drug discovery teams. RushData was built to help AI and machine learning teams move from digital antibody sequences to actionable experimental datasets. RushData is an integrated, high-throughput platform combining one-day CHO expression, rapid binding analytics, and early developability profiling to generate actionable datasets for antibody discovery and optimization. Designed for AI and ML-driven workflows, it enables faster, data-rich decision-making at scale. The RushData platform can support up to 3,000 antibodies in a batch, making it suitable for large candidate panels generated through AI antibody design, and allows researchers to identify liabilities early with developability assessments to reduce late-stage failure and downstream risk. To learn how RushData can support your antibody discovery program, visit BioIntron's website, www.biointron.com, or contact the BioIntron team at info@biointron.com.